Okay, we're gonna settle in for our last 45 minutes together and do some reproductive physiology. We're not going to cover every single slide in this slide set because it's just too much, but this was like a 500 slide set that I chunked it down to 72, I think is what I got it to. So I really tried to like high yield as much as I can. It might be 68. I don't remember how many slides I ended with at the end, but it was like 500 or more to start with. So I really tried to like focus in for y'all. But we're going to start a little bit with some combined reproductive physiology, and then we'll move into some specific male versus female reproductive physiology. All right, on screen shared, we're recording. Okay, great. So in the male and female system, there's the same 4 tasks that happen in both. When we're talking about development, we're talking about creation of gametes, we're talking about creation of our sperm and our ova or our eggs. So we have to form specialized cells for sexual reproduction, our gametes, sperm, male, ova, female. We have to bring those gametes from male and female together through intercourse or coitus. We have to combine the genetic information then from a maternal and paternal component or a maternal and paternal gamete through fertilization. When egg and sperm fuse, that creates our zygote. The zygote, you could say, is our first stem cell, right? Our pluripotent stem cell that can become every type of tissue. It's non-differentiated yet. And that's going to support the development of our fetus, gestation, or baby parturition. So where we get our GPs, right? If you've ever seen the patient where we have the gravidan parus and we have the G1, P1, et cetera, et cetera, et cetera, that's from these words here. Okay. So a reminder, all of this control starts all the way up top in the brain and the endocrine system. Our hypothalamus to our anterior pituitary, to the gonads, ovaries, testes, to our target cells or organs. GnRH is the communicator between hypothalamus and anterior pituitary. LH and FSH from anterior pituitary to gonads. And then from gonads, we release sex hormones, depends on the sex, depends on the gonad, and inhibin. Inhibin's a new one that we haven't discussed. It's released from both testes and ovaries, and it exerts negative feedback on FSH specifically from it being released from the anterior pituitary. So we'll kind of see how that comes into play a little later on when we talk about development. But that's the one that's new in this slide. So when it comes to male hypothalamic pituitary gonadal control, that's your HPG axis. We often see as HPO, but males also have gonads, so we need to say HPG. So FSH is what's going to stimulate spermatogenesis, the creation of sperm. It's going to do this indirectly by stimulating cystinocytes or cystentocytes to release androgen binding protein, ABP. ABP essentially keeps testosterone high or elevated near cells that are spermatogenic, trying to create sperm. LH will bind to interstitial endocrine cells, and it will encourage them to secrete more testosterone, and increased testosterone that increases over time triggers spermatogenesis. Testosterone also will enter into circulation via blood and will stimulate our sex organ maturation and our secondary sex characteristics and libido. And it will also exert negative feedback on the hypothalamus to inhibit GnRH and inhibit our anterior pituitary hormone release. Inhibin, like I mentioned, is released as well from our organs or our gonads. In males, it's released by the cell cystentocytes in the testes when sperm count is elevated, meaning that it doesn't need more sperm. There's plenty of sperm, so we can stop spermatogenesis. And so it's negative feedback. Okay. So big words here are the ones highlighted on this page. We'll define some of these cell types here moving forward. Why does this not happen initially? Why does this happen at puberty, right? GnRH is suppressed before puberty due to low steroidal sex hormones like testosterone. There's low circulating levels initially. As the reproductive organs grow and as puberty approaches, the hypothalamus essentially becomes less sensitive to inhibition. It becomes more likely to bind and to respond to testosterone. So testosterone will slowly start to increase because these sex organs are getting bigger and more mature. And then the hypothalamus, it's essentially negative feedback, is going to become less and less sensitive. So it's more likely to respond to the need to produce GnRH. So both are occurring at the same time. It will then eventually start to release, once that threshold's met, it will start to release GnRH in a pulse-like manner. The anterior pituitary can then respond, and that whole cascade occurs. But that's why it doesn't happen initially. We have to have the sex organs start to mature, start to grow, start to develop. We have to have an increased supply of circulating testosterone, and we have to have that happen, and we have to have a less sensitive hypothalamus that's essentially saying, I don't need to have as much negative feedback. I can be more responsive. The hypothalamus threshold for inhibition continues to rise. It'll lead to more and more sex hormone released until the adult pattern of hormone interaction is achieved. So this is what I'm talking about. This is testosterone. So initially you'll have this like little blip, and then during fertilization, that's essentially what will differentiate into male or female fetus. Then this will decrease at birth, and a tiny little blip here, and then it stays pretty low until it reaches puberty, and then it spikes in adulthood, and then over time it'll decrease. And that's your natural kind of cyclical nature of testosterone. So you could say there's 3 spikes: one during fertilization, when that differentiation happens, one right after birth, and then one at puberty into adulthood. These are secondary sex characteristics for males. If you don't know, I listed them here for you. But now female control. So puberty for females. FSH and LH are elevated at birth for females, but then they drop and they will remain low in your pre-puberty years. Your puberty years for females is the same. Once your reproductive organs have grown to adult size, and then they can become functional. It will occur in response to, just like males, a rise of gonadal hormones, estrogen and progesterone. You'll see secondary sex characteristics to appear, and that's will be the earliest time that reproduction is possible. We'll kind of see this cyclic nature occur a little bit later when we talk about our menstrual cycle and uterine cycle and ovarian cycle. At the end, right, the end stage of development, so we have birth, we have puberty, and we have menopause. You would call it menopause once menses have ceased for an entire year. There is no equivalent to menopause in males. This is female specific. Males can continue to produce sperm their whole life, much well into the 8th decade and beyond. But for females, declining estrogen levels can have a whole host of different symptoms and responses. But obviously the loss of menstruation, loss of ovulation is the most stark contrast we'll talk about here in this slide set. Okay, that was our general preview. Now we're going to do a little genetics, a little bit. So meiosis. We haven't looked at this since general bio. Okay, yeah. So meiosis terminology, just to get us on the same page. There will be questions on this on NPLEX in some way, shape, or form, often in the guise of oogenesis or spermatogenesis. So that's why it's important to review. So we have our sister chromatids, our homologous pairs of chromosomes, and our non-sister chromatids. Sister chromatids, there's 2 per chromosome, and they are identical and joined by a centromere. Versus a non-sister chromatid, it's within a homologous pair, which we'll define chromatids of 1 chromosome with a non-sisted chromatid to the other chromosome pair. So they are not identical, they are different, but they're hanging out in a homologous pair of chromosomes. This is how crossover can occur. Non-sister chromatids can cross over in meiosis to share genetic material and increase genetic variability. This is why we all aren't identical. Homologous pairs of chromosomes, 1 member of each pair is from a paternal chromosome, 1 from the maternal chromosome. They look alike, meaning that they code for the same genetic stuff, the same genes, the same traits, but they won't be identical unless if you had 2 identical twins mating, which would be unfortunate. But they will not be identical. They will be close, though, in that they will match the same type of chromosome, the same type of gene, chromosome 1 and chromosome 1, chromosome 21, chromosome 21. Those will be homologous pairs, 1 from the maternal legion, 1 from the paternal. Some more terminology. This is where people start to like block out words. So diploid and haploid, okay? Diploid is represented as 2N. It's 23 pairs of homologous chromosomes, or 46 total, right? So human genome, we have 46 chromosomes, 23 pairs. In trisomy 21 in Down syndrome, we have 47. because we have an additional chromosome. So instead of 46 chromosomes total, our 21st chromosome pair has 3 chromosomes in it instead of 2. It's not a pair, it's a triplet. And so that's how we end up with 47 in that pathology. But in normal diploid state, we have 23 pairs of homologous chromosomes. The chromosomal number represents the normal number in most body cells. You have 2 sets of chromosomes, 1 from each parent. Haploid number is represented as N. It's 23 chromosomes total. So it's the single chromosome on its own. And this would be like the number of chromosomes in a gamete, in a sperm, or in an ova or egg. Gametes have only 1 member of each homologous pair. So either a maternal, egg or ova, paternal, sperm. When 2 haploids meet, they form a diploid, which we call a zygote if they're actually fertilized, right? So egg, haploid, sperm, haploid, fertilization, diploid, okay? I know it's kind of basic, but we see these words without context, and it's like we trauma block them, I feel like, and forget everything we know about meiosis. So I want to just lean into the definitions. If you can lean into these 2 slides, you can probably answer majority of these genetic questions on meiosis, mitosis. Okay, so know those first. So the overview, what is our goal of meiosis ever, right? The goal is to reduce the number of chromosomes in our gametes by half so that the zygote, when both gametes combine, doesn't have twice as many chromosomes. So meiosis is essentially saying, like mitosis, right? It's just exact replication. We replicate, we divide, we have 2 identical cells. That's mitosis. It's what the majority of cells in the body are doing. In meiosis, we want to replicate, we want to divide, but we don't, when we have an egg and a sperm, we don't want to have an egg and a sperm that already have a full chromosomal pair. We also will have duplicate of everything in the body, and that would not be compatible with life. So our 3 kind of steps of this process involve interphase, meiosis 1, and meiosis 2. In general, interphase is when we are replicating. All chromosomes get replicated. DNA replication occurs. It occurs only once. So in this interphase stage, for spermatogenesis, oogenesis, all these processes, there's only 1 time where DNA is replicated initially. That's in interphase prior to meiosis 1. During meiosis 1, the homologous pairs, so we see we have a pair of homologous chromosomes. Now we have the pair of replicated homologous chromosomes, both 2N. Then in meiosis 1, we have 2 cells where these homologous chromosomes have separated. Okay, we call these haploid with a replicated chromosome because we technically only have 1 pair. We have the green pair here and the purple pair here. But because we replicated and created an identical duplicate of the green one, we have 2 identical duplicates of the same chromosome. So this is considered haploid. We just have double the amount of DNA. Okay, this is considered a reduction division of meiosis because we've reduced from 2N diploid to 1N haploid. We then go meiosis 2. In meiosis 2, these sister chromatids here, these identical chromatids, they are going to separate into individual cells that are all haploid cells. These would all be represented as like ova or ovum or sperm. In oogenesis, we'll see that we don't create 4 identical haploid gametes at the end. We only get 1 in this process because it's a little bit more complex. But in spermatogenesis, we will get 4 identical sperm. Okay, this is an equational division of meiosis, meaning that we went from haploid to haploid. We did not change. So diploid to haploid is meiosis 1. Haploid to haploid is meiosis 2. This same process occurs with a little bit of variation in flare in both spermatogenesis and oogenesis. A couple important things to know. A tetrad, this is just looking closer at meiosis 1. We're not going to go through all this, but a tetrad is formed here in this first kind of prophase stage of meiosis 1. A tetrad just means it has 4 chromatids present. So it's both the chromosomes with their replicates. So a tetrad is just all 4 of those chromatids present prior to having them separated. Meiosis 2, this looks pretty much like mitosis, except for there's no replication at the beginning. And the cells at the beginning are haploid. So meiosis 2, you could say, is pretty much like mitosis. It has a prophase, a metaphase, an anaphase, and a telophase and cytokinesis. Okay. That's meiosis. That's our overview of that. I want to, okay, yes, please.
Later, but so we talked about oogenesis. It first the mitosis to get to the primary oocyte and then meiosis.
We will do, you'll see there'll be a replication at the beginning, and that would be technically a mitosis. And then you go through meiosis. We're gonna look at it. I don't want to state it before we get there. That's okay. We're gonna get there, I promise. That's my main goal. I have on these pages, I'm not going to read them to you, but what I've tried to do is for the major organs or components of male and female reproductive system, I've tried to highlight like these are things you should definitely know or identify in these regions. This is scrotum and testes. So I'm not going to read these, but these would be high yield things to try to commit to memory in these structures. And again, I do have some pictures, so apologies if that's like triggering. The pathway of sperm. We are going to talk about spermatogenesis, so I do want to talk about the pathway of sperm. So seminiferous tubules in the testes is where sperm is going to start. We will see this is where creation of sperm occurs first. It's going to flow through the straight tubule, the testes, the efferent ductiles, and the epididymis. Once we're to the epididymis, it's another pause phase. That's where sperm mature. After the epididymis, it's going to move through the accessory ducts into the ductus deferens, ejaculatory duct, urethra. Accessory glands like seminal glands and prostate are going to help create the fluid for sperm to live and for sperm to move through after ejaculation or during ejaculation when that occurs. Specifically, the seminal glands produce fructose. Fructose is what sperm consume for energy. That's their sugar they're utilizing. The prostate has PSA, but it also has some slightly acidic fluid that helps activate sperm. So both these fluid kind of sources are helping with the sperm once they're released, be able to actually then migrate to their goal site. And there's your testes structure. If you're visual, you can kind of walk yourself through what these structures are, starting all the way from your seminiferous tubules and then moving all the way up into that spermatic cord. Males, they are interesting. They have three regions of the urethra. So again, another anatomy structure that would be high yield to know the different portions of the urethra in males, what they're called, and where they're located at. I would go from deep to external. Okay, so spermatogenesis. We'll go through this process. This is kind of your overview picture from the spermatogonium, which are diploid, and then moving into your spermatocyte, so on and so forth. So we're going to look at this process as a whole. The key players, it's located in the seminiferous tubules, spermatogenesis. It happens, the timing is puberty. Spermatogenic cells are the cells that give rise to sperm. And there's three major steps. This is when we kind of get into the mitosis conversation that you were asking about. So mitosis of spermatogonia occurs first. This is your stem cell. It forms two spermatocytes. Then you undergo your meiosis 1 and 2. Your spermatocytes form secondary spermatocytes, which form spermatids. And then spermatogenesis, spermatids become sperm. And we can see that represented here. So this is our mitosis phase. So that's where we're essentially undergoing normal cell replication. We have meiosis 1 and 2. We have our spermatids here, and then they're going to mature later on into mature sperm. Okay. The important cell type that's really helping this process occur is our cystenocytes or Sertoli cells. You could see both used. Sertoli is much more the common cell type, but I've seen both on the NPLEX. So just put them together in your brain. These are large cells. They surround the developing spermatogonium. They essentially divide the tube into two different compartments, a basal and a luminal compartment. The basal compartment is where the spermatogonia and the primary spermatocytes are located in these cystinocytes or Sertoli cells. The tight junctions here form the blood-testes barrier. Sperm is not around during fetal development, right? It starts at puberty. So if sperm got into circulation, blood circulation, the body would identify it as not self and would target and attack it as a foreign invader. So the blood-testes barrier is really important to prevent sperm antigen or sperm themselves from escaping into the blood and causing that auto attack. The Sertoli cells have a ton more functions besides kind of these like barrier pieces. They do produce two mediators to regulate spermatogenesis, androgen binding protein, which keeps testosterone levels high to stimulate this process, and inhibin, which can inhibit this process by inhibiting FSH release if there's too many sperm present. So these cells are really the controllers of spermatogenesis. They can increase it or they can decrease it using hormones. Okay. So the first step I mentioned is the mitosis of spermatogonia. This is how we form spermatocytes. Spermatogenesis, this starts at puberty. We start with spermatogonia. These are stem cells. They divide by mitosis. At puberty, you'll get two cell types, type A and type B daughter cells. Type A daughter cells remain at like the germ cell, the stem cell. They continue to divide. Type B daughter cells move towards the lumen. These are going to be the ones that develop into primary spermatocytes. So mitosis occurs first. We get spermatogonia, two primary spermatocytes, and we have essentially two daughter cells that are formed, one that stays this germ cell, this stem cell, the other that will develop into the primary spermatocyte and eventually becomes sperm. Meiosis is where we take our spermatocytes to spermatids. It's also where we go from our 2N to our N, our diploid to our haploid. So meiosis 1, we have a primary spermatocyte, which is diploid, and we will get two secondary spermatocytes, which are haploid, two N's, N plus N equals 2N. That's how I remember that. Meiosis 2, we have a secondary spermatocyte that becomes a spermatid. So one spermatocyte, one secondary spermatocyte, one spermatid. So we technically could get 4 per cell. Spermatids are small, round cells with large nuclei. They're found close to the lumen. Nearly all the genes at this point are turned off. DNA is just compacted into these pellets. And then there's step 3. This is the longest step. It takes 64 to 72 days, so a while, to a little over 2 months. This is where we get spermatids to sperm. This is the maturation of spermatids, or the maturation of sperm. Spermatids contain the correct haploid chromosome. They are haploid, 1N. They are non-modal in this stage. During spermatogenesis, this is essentially where the spermatid elongates, loses cytoplasm, and forms a tail. And this is where you become, once you go from spermatid to sperm, you'll see the 3 major regions, the head, the midpiece, and the tail. So that's through this last stage. So mitosis, meiosis, spermatogenesis. Okay, this is the process you'd see. Okay, how is our male repro in 20 minutes? The same with female. Again, some anatomy pieces. We already saw this questioned. One to note, there's a suspensory ligament of breast tissue. This adheres or attaches breast to underlying muscle. There's a suspensory ligament that anchors the ovary laterally to the pelvic wall. So depending on your area, there's a suspensory ligament for both. Okay, you'll be able to get context clues from the question, what it's asking about. But these would be the ovaries that would know. The suspensory ligament plus the mesoovarium are the broad ligament. So these two together are the broad ligament. These are often, I would say, arguably, potentially always tested on the NPLEX in some way, shape, or form. Ovaries, again, some important anatomical structures. The gametes are formed in the outer cortex of the ovary. There is an inner medulla that has blood vessels and nerve supply, but the outer cortex is where our follicles are located and where gametes will form. These are the locations of the uterine tube that you should know, 3 different locations. The main one to know would be the ampulla. That's typically where fertilization occurs. Okay. This is a picture, if you are visual like me. Shows the different ligaments, it shows the different structures. Okay. Again, more supports of the uterus. These are just more ligaments, more supports. This is anatomy. I'm not going to go in depth. And then the uterine wall, the 3 layers. The most important piece about the uterine wall is to understand the endometrium, because the endometrium is where implantation will take place, right? So that's an important functional layer, structural layer to know. The stratum functionalis, the functional layer, has changes in response to ovarian hormone cycles. So it will change to what the ovaries are secreting. This is what you shed during menstruation. The stratum basalis is the basal layer. This is where you can form a new stratum functionalis. These are like the stem cells. It does not get shed. It does not respond to ovarian hormones. That's why you don't have like your full endometrium doesn't shed every time you menstruate. You have a piece there left. Blood supply, lower yield than abdomen, brain, heart, still commonly tested on. Okay, now our exciting event, oogenesis. So here's your answer. Yes, mitosis does occur at the beginning. I just didn't want to get us there before we got there. But I do need to remind you that when we talk about oogenesis in female reproduction, we also need to think about follicle development at the same time. But they're separate processes occurring together at once. So we'll be kind of talking about them in tandem of what's happening in order. And so I will try to kind of clarify, this is the ova that we're focusing on, oogenesis. This is the follicle that's supporting said ova, okay? So this is our overall process where we're going. The three main differences between male and female gametogenesis, the production of primary oocytes occurs only in the fetus. It's different than males. They can produce their primary oocytes or their primary spermatogonium to primary spermatocytes all throughout their life. In the primary oocytes, meiosis is arrested in late prophase 1 and resumes only years later, if at all. And in secondary oocytes, meiosis is arrested in metaphase 2 and is only completed if fertilization occurs. So there's some changes. We have these arrested stages because we're born with all of our primary oocytes in theory at birth. And so you can see kind of some comparisons between the two of the gametes. If you think about how the number of gametes per lifetime, women less than 500, men 1 trillion or more. It's like, it's a very stark contrast. That's why conception is such a crazy thing to think about. Okay, starts in the fetal period. So we're going all the way back to embryology, but not. So oogonia are diploid stem cells. There are spermatogonia. They both have gonia. These divide by mitosis and they produce our primary oocytes. These undergo meiosis to produce our secondary oocytes. These undergo meiosis 2 to produce ova. So that's your kind of following process. Oogonia, primary, secondary, ova. All right, so at least follow there. At birth, females are presumed to have a lifetime supply of primary oocytes. So this is where we are at birth. Our follicles are related to our oocytes, right? There's a relationship between the two. The ovarian follicle is like the functional unit of the ovary. It's in that outer layer. It encloses a single oocyte, and there's two kind of cell things. There's pregranulosa or granulosa cells. It's pregranulosa cells that surround that oocyte if it's only one layer. It's granulosa cells if there's multiple layers. So essentially, the bigger the follicle, it's probably granulosa cells. If it's only one layer of cell around it, it's called pregranulosa cells. These are just the cells that line or surround a single oocyte in an ovarian follicle. Follicles also have stages of development. It starts with primordial follicle, which is great. Primordial ooze, primordial follicle, you start there. That's a single layer of pregranulosa cells surrounding a primary oocyte. So this is kind of where we're at birth. Follicles then can become primary follicles. They surround primary oocytes. Secondary follicles surround primary oocytes. And vesicular, antral, or tertiary follicles, they have an antrum, and they also can surround primary oocytes prior to ovulation. A primary oocyte inside this type of follicle, tertiary follicle, can resume meiosis and become a secondary oocyte. So the follicle has to get here prior to the primary oocyte going to resume and undergo meiosis. So that's why this is a relationship between the two. There's two fates of your follicles, atresia and ovulation. Atresia is apoptosis, programmed cell death. The majority of follicles are never recruited. The oocyte and the surrounding cells all die. Ovulation, every month during puberty or after puberty, around the time of ovulation, you'll select a few primary oocytes. They're activated. This is caused by high FSH levels or other hormone levels, but especially FSH. And from this activated group, one will be the dominant, one will win. That's the one that gets ovulated every month. Typically the one that's the most dominant is the one that's the largest, has the most layers, the most antrum, the most support, or is the healthiest. Follicles and oocytes can take up to a year to mature before ovulation can occur. You have two kind of phases. Phase one, you have your gonadotropin independent phase, it's preantral. It involves paracrine signaling, local signaling. Phase two is your FSH-LH signaling. That's once you have your anterior pituitary activated post-puberty, right? That's when your dominant follicle is selected and your primary oocyte resumes meiosis 1. So if we look at these events together, we can kind of follow them down. This is before birth, throughout life until menopause, from puberty to menopause. We can see that we have our follicle development, our pregranulosa cell, our oocyte, and our primordial follicle. Our primary oocytes here as well. This is like at birth. Our primordial follicle can become a primary and a secondary follicle. We still see we're still primary oocyte this whole time. We've not changed. From there, once we have a vesicular antral follicle, we'll see meiosis being completed, meiosis 1 in the vesicular follicle just prior to ovulation. And then once ovulation occurs, if our oocyte, essentially our secondary oocyte, becomes fertilized, then meiosis 2 will complete. But if this secondary oocyte does not become fertilized, if it does not meet sperm, it will stay arrested in metaphase 2 and under meiosis 2. And it won't finish meiosis 2 unless if it actually has a sperm that's penetrated the oocyte. So that's kind of the kicker for maturation. Okay. One other note that's interesting about oogenesis is when we saw our spermatids, we saw they were all equal, multiple spermatids that look the same. When we go through meiosis 1, you'll get a first polar body that's haploid, but it has a small amount of cytoplasm and it's a essentially non-capable oocyte. It does not become a secondary oocyte, essentially becomes degraded. So we only get, from our meiosis 1, we get a first polar body that becomes nothing, will degenerate, and then a secondary oocyte. Our secondary oocyte, if it goes through metaphase 2, if it finishes meiosis 2, we'll have an ovum, and our second polar body also will become degenerated. So that's kind of our process here. I have the words, if you're like wanting to read through it. I find that picture to be a great place to start, because I really think if you can understand this picture, you can understand this process. You don't necessarily need to have to read all the words that explain what's happening. Okay, great. All right, great. Few more things and then we'll let you go. 10 more minutes, you're almost there. Then we do biochem tomorrow. Yeah, let's go. Oh yeah, it's gonna be so great. I'm gonna be highly caffeinated. It's gonna be super fun, jacked up on coffee. We draw on things. Yeah, crazy day. Okay, follicular phase. So there's two phases. We're looking at the ovarian cycle. I just want to kind of define a few things, and then this is available for you. We have our follicular phase. During this phase, this is when we select our vesicular follicles to become sensitive to FSH and allowed to grow. The dominant follicle will be selected during this phase, and your primary oocyte of your dominant follicle will undergo meiosis 1 to form a secondary oocyte and first polar body. We then have ovulation, where you can see Mittelschmerz pain. That's the twinge of pain sometimes felt at ovulation by some women. So if you feel pain with ovulation, you can say, oh yeah, it's just my Mittelschmerz. Your ovarian wall at this point, your ovary wall at this point will balloon out from this massive follicle that's been dominated by FSH and growing. And that's when you will expel your secondary oocyte with its surrounding corona radiata into the peritoneal cavity. Ideally, it gets taken up by your fimbriae and moved into your uterine tubes. If it doesn't, and it happens to meet sperm that process, that's how you can get ectopic pregnancy actually in the peritoneal space. More rare, obviously, but can occur. Every once in a while, you can have more than one secondary oocyte released per ovulation, which could lead to fraternal twins. Identical twins are from one oocyte, and then the daughter cells are separated. Sometimes we'll look at how this is released, but sometimes hormones can dictate it. They should. Sometimes we don't respond to them. And that's when you can see certain pathologies. The luteal phase after ovulation, your ruptured follicle collapses, your antrum fills with clotted blood. Your granulosal cells will form the corpus luteum. They'll secrete some progesterone and estrogen. If no pregnancy occurs, this will degenerate in about 10 days. And then you kind of move into your bleeding phase, your first day of your menstrual cycle, your first day of your period. If pregnancy occurs, though, the corpus luteum will produce hormones like progesterone that sustain pregnancy until your placenta is grown enough to take over around 3 months, right at the end of the first trimester. That's when placenta takes over hormone control. So to establish this cycle, we have essentially that hypothalamic release of GnRH. It releases it pulsed like it does with males. So that's the same. And that's what helps give us our actual ovarian cycle. This occurs until menopause. These are the hormones involved. We've kind of talked about FSH and LH, their jobs. FSH is helping the granulosa cells to release estrogen. LH will prod thecal cells to produce androgens, which then local tissue converts to estrogen. So it's all about the estrogen here. We also have some positive and negative feedback loops. The negative ones we've discussed a little bit already with inhibin and also increased estrogen. But positive feedback can also occur. This is specifically for your LH surge. That is going to trigger what? Ovulation. So this is where we see a short positive feedback loop set in a negative feedback cycle. Estrogen will continue to rise as a result of the continued release by the dominant follicle. It's producing its own estrogen. When the levels reach threshold, the brain and anterior pituitary have a brief positive feedback loop where you have this huge release of LH due to an increased localized secretion of estrogen into systemic circulation. Once that LH surge happens, it's this huge rush of LH, then that will provide negative feedback. So that's why LH doesn't continue to increase forever and ever. It has this big bump, and then negative feedback kicks back in, and it stops GnRH from being released. But that's our surge that triggers ovulation. The ovarian cycle is talking about the ovaries, but this process, you can also think about the uterine cycle, the menstrual cycle, right? The ovarian cycle is what do we have to do to have ovulation take place? The uterine cycle is our typical menstrual cycle that we think about with menstruation. And it's really focusing on our endometrial change. What's the endometrium doing? 1 through 5 is our menstrual phase. 6 through 14 is our proliferative phase before ovulation. And 15 through 28 is our secretory phase or our post-ovulatory phase. During your menstrual cycle, during menstruation, ovarian hormones are low. Gonadotropic hormones are starting to rise. Stratum functionalis will detach and shed. That's how you have menstrual blood. And by day 5 to 7 to 8, depending on the woman, the female, the person with uterus, by day 5, the growing ovarian follicles will start to produce more estrogen. And so that process will eventually stop the bleeding, stop menstruation. Days 6 through 14, again, this is a 28-day cycle, so kind of keep that in mind. Not everyone has a 28-day cycle. Actually, in fact, most women do not. But days 6 through 14, rising estrogen levels will generate a new stratum functionalis layer, that thickened external layer of your endometrium. As that thickens, there's more glands, there's more arteries, and you'll also see more estrogen. Estrogen will increase synthesis of progesterone receptors in the endometrium. We're essentially trying to prepare this space for implantation during this phase. We're thickening it, we're getting progesterone receptors ready, we're like getting our body ready. They'll also start to see some thick, sticky cervical mucus to try to help sperm pass along, because the uterus's goal is to implant a fertilized embryo. That's its only job, right? Is that and kind of helping with regulating blood flow in that region. Once ovulation occurs at the end of the proliferative phase around day 14, if it implants, it'll implant 7 days or 6 days later, right? On day 6, it sticks. So 6 days after ovulation, it'll implant, but it'll fertilize much sooner than that. The last phase is typically the most consistent in duration physiologically. This is not talking pathology-wise. The endometrium is getting ready for that embryo to implant. You'll see increased progesterone levels from the corpus luteum. So you'll start to see more gland secretion, more nutrients present. Again, it's like, yes, here comes the embryo. And then if that doesn't occur, it's like, oh no, and it degenerates and sloughs off and the process starts again. Spiral arteries are the arteries that spasm and kink and can eventually cause the hemorrhage and bleed that we associate with menstruation. If you're visual, this kind of showing you the LH and FSH surges, kind of what's going on with your follicles, what's happening with estrogen and progesterone, and then what's happening with your uterine layer. I find this, like my oogenesis picture, the most helpful picture for understanding the relationship between hormones, the ovarian, and the uterine cycle. If you are a table person and want to know what estrogens, progesterone, and testosterone are going to do in the body, here's your table for you. And then all of that is done with 1 minute to spare. Any questions on repro stuff? Repro phys? All right. Well, thanks for listening today and hanging out. We will be back tomorrow. It'll be all biochem the whole day. Bring your biochem questions. I'm going to post some worksheets. I would bring markers or colored pencils. I'd bring paper. Expect yourself to be drawing and writing for the majority of the day. So there we go. Thank you.